Well, thank you, Marty, and all the SCOR group members for inviting me to give this short talk. The aim of this talk is just to provide like an overview of what has been used so far to be able to estimate or even measure the biological consumption rates of the methylated sulfur compounds. And I took this nice diagram that Frankie and others put together last year. And of course, it all starts with the MSP synthesis, which is something that will be touched in another, will be dealt with in another of this series of workshops, I think on the 3rd of October. But after the MSP synthesis, there is this consumption or turnover of essentially particulate or total DMSP, which also will be dealt with in another workshop on the 10th of October. But let me give just a brief idea or a brief overview of how we have been measuring the turnover of total DMSP in seawater, because this is the first channels towards the other compounds. And back in 2000, we already proposed a very rough method, actually very coarse, which is just putting a bottle of unfiltered water in the dark. And then with the assumption that the MSP synthesis is arrested in the dark, which is kind of controversial because there are some indications that it can resume in the dark, but essentially it is essentially true that most of the DMSP synthesis is in the light, at least for the few works showing that. And with that assumption that you stop synthesis in the dark, then of course, as you see, the concentration of total DMSP, this is important, the total DMSP going down, okay, this is total consumption by any means. And since it is total, this means that it addresses not the conversion of the MSP, not the release of the MSP into the MSP dissolved, but the real consumption of whatever pool of the MSP, so the transformation to something else. As I say, this is a very rough method with lots of potential drawbacks. If you want to use this, be sure that you use as big a volume as possible, because the other assumption is that the bottle contains the entire community, whatever it means, microbial community, so including grazing going on and things like that. And then for a relatively long time, you see this decline in the concentrations. Here you have 3 cases with low DMSP concentrations, total DMSP concentrations, medium and very high. And indeed, this happens. Look at the open triangles only here. And of course, another assumption for this is that the loss of the MSP is not light dependent because you put the sample in the dark. There is one problem with this. By experience, I can say that there is one problem, that if the sample comes from the light, you are around midday, you take a water sample, you put it in the dark to follow the DMSP decrease. Very often you have first an increase in the short term of the total DMSP, and then it goes down. This is probably because this synthesis of the DMSP is not immediately arrested, and because you are incubated in the light, in the dark, but coming from the light, there is still some inertia for DMSP synthesis. So that's a problem. And of course, then you can take it from that time on as a consumption. Well, this is very rough, but there are not many other ways of having an idea of how much the MSP in total turns over every day or every hour. Then, of course, we are very interested typically in the consumption of the MSP dissolved, how this can be addressed, how this can be approached. And one way is to use the competitive inhibitor method. Any use of competitive inhibitor, the way it works is by adding a compound that the microbes say take it as your target compound, and this way you stop the consumption of your target compound. This is why it's called a competitive inhibitor. It's not killing the microbes, it's not inhibiting the microbes, it's just making them get confused by the inhibitor so that they are not busy in the consumption of your target compound. In the case of the MSP, Ronkin and Gerard already published in 95, this method where the addition of glycine betaine would block, in the short term, would block the consumption of dissolved MSP. And the assumptions are that these two compounds share the same transporter in bacteria, which was then confirmed by Ronkin and others in 98, and even by phytoplankton, which we confirmed in 2006. So the idea is that they share the same transport, so you kind of saturate the transporter with another compound, in this case, glycine betaine. Note that this only works at a very high GBT concentrations, even though we know that the dissolved DMSP concentrations are low. You need to add orders of magnitude higher concentrations of GBT, and it only works in short term, less than 6 hours. After a while, the inhibition is not so efficient. And the pros are that these are very convenient, or the only way to estimate the MSP consumption, the dissolved MSP, or one of the only ways to estimate the MSPD consumption when radioisotopes cannot be used by any reasons like regulations or whatever, danger, etc. Daniela will talk about the use of radioisotopes. Then we can do a similar thing for DMS. So again, use a competitive inhibitor method, and Wolf and Keen already tried a full list of different compounds for their efficiency in blocking or inhibiting DMS consumption, bacterial or microbial DMS consumption. And I also tried several compounds some years later. And for instance, one that was nicely inhibiting DMS consumption, and there is no surprise here, is dimethylselenide, because it's so similar to DMS, to dimethyl sulfide, that even at rather low concentrations, nanomolar concentration, it really nicely blocks DMS consumption. The problem is that it's not super friendly to use because it's a volatile, and then preparing a solution with a volatile without the use of other compounds like glycerol or things like that, it's always a pain. If you use more like a general harmful substance for bacteria like chloroform, yes, you block DMS consumption as well. So you see an accumulation, but you overestimate this accumulation. So you're probably killing algae, releasing DMSP. I mean, you're kind of enhancing the formation of DMS. So it's not convenient. It was shown not to be very convenient long ago. So another one that works very nicely, and that's the one that we've been using over the last 20 years, is DMDS, dimethyl disulfide, which is taken as DMS by the microbes so that it blocks DMS consumption. Typically, we use it at concentrations of 200 nanomolar. The assumption is that the MDS inhibits completely microbial DMS consumption. This has not rigorously tested yet that it's completely. It seems that it blocks most of DMS consumption indeed. I mean, here on the left, you have an example of two samples from the Arctic. This is by Marty long ago doing his PhD. And you see that when you have the net change in filled circles or squares without any addition, net change of DMS over time. And then when you add the inhibitor, you have a faster accumulation, meaning that you have inhibited consumption. Sometimes this accumulation is not almost any faster than the net change, so there was no extra production, or there was no consumption indeed. But sometimes this is very clear. You have to add at least 10 times higher concentration of the inhibitor, DMDS in this case, than the endogenous DMS concentration. So we typically use 200 nanomolar. And sometimes to see significant difference between the two curves, it requires long incubations, long meaning longer than 8 hours. So it's not for very short term. And if you do it in the light, this is always a problem. For instance, when we're looking at how DMS production and DMS consumption varies over the yield cycle, of course, you have to correct for photochemistry, for photochemical loss in your bottles that is occurring along with these biological transformations. It's appropriate again when radioisotopes cannot be used. And the problems, the cons are that higher concentrations higher than 200 nanomolar in our experience enhance photochemical DMS loss. So never use concentrations higher than 200 nanomolar in the light, especially. And the other thing is that the MDS is a bit of a pain. It sticks to surfaces. It gives you some memory in the chromatograms. And if you are doing very fast measurements of DMS by GCFPD, and then you add the MDS, then you have to elute this DMDS that you added, and so you have to wait for longer, and this is not that fast anymore. I didn't say that this accumulation over time with inhibitor is what we call gross DMS production. So the production because we blocked consumption, and the difference from the net change would be the DMS consumption. So you get the two things. And then if you want to approach dissolved DMSP transformations, then so far the only way we have is to use the stable isotope method, tracer method. And here you have an example. This has been developed essentially, or among others, but essentially by Tortel and Philippe Tortel and John Dacey's groups. And here, for instance, by adding, the idea here is that you add a labeled, stable isotope labeled compound, and you follow its loss over time, and you can even follow the product, the accumulation of the product, so that you not only know the loss rate, but the fate of this compound as it transforms it into something else, because you can discriminate them by mass. And here you have one example down here, which is by adding 13 carbon labeled DMSO, and then just monitoring the appearance of 13C labeled DMS, you have you can estimate the transformation or the reduction of DMSO into DMS. By doing a similar thing with the MSP, by adding deuterated DMSP and then monitoring the appearance of deuterated DMS, you can we can estimate the cleavage rates, or by adding the deuterated DMS and then looking at its loss, then you estimate DMS consumption, and this would be the endogenous DMS net change. This is pretty powerful. The assumption is that the tracer that you're adding, the labeled tracer that you're adding, behaves the same as the endogenous compound. So you need to add this tracer concentration at very low concentrations, and these authors, they say less than 20% of the endogenous concentration of DMS, for instance, or DMSO or DMSP. You need to correct for, or at least to watch out for, any unlabeled compound that is present in your labeled compound solution, because there's nothing like 100% purity in these labeled compounds. And attention to potential interference with other compounds with the same mass as your tracer. And the pros is, again, when you cannot use radioisotopes, you can go for this type of, this is harmless, and you don't need special facilities for doing this, and provides, as I said, provides the rate of transformation, but it also allows for tracking the fate of these compounds as they transform into other compounds that you can also measure. The cons is that, okay, of course, it's not based on retention time, it's based on mass, so you need a mass spec. And that's it. I only want to finish saying that this isotope, stable isotope tracer method can be applied potentially to DMS, as I said, but also to methane thiol, for instance, that we haven't addressed yet the transformation process rates for methane thiol. Why not using this stable isotope tracer? And I think that's it. I mean, food for discussion. I hope I was short enough.
Thanks a lot, Rafael. It was very interesting, and now I realized I didn't introduce you properly, although probably you don't need much introduction here.
No worries.
But for those who don't know Rafel, he has been making like extensive contributions to the study of these super compounds from the point of view of methods, measurements, concepts, and modeling for the last 30 years. So now it's time to introduce Daniela del Valle, our next speaker, who will talk about the radio tracer isotope method. And Daniela also has extensive expertise using these radioisotopes for measuring the fate of DMSP, but also DMS and DMSO. And well, this comes from her PhD and with Ron Keane and in Ron Keane's lab. And Daniela was also the co-chair of DMS Pro, so we are very happy to have her here today. The floor is yours.
Thank you. Okay, let me share my screen and arrange things here. Okay, great. Well, hello, everyone. It's really nice to have this turnout for the workshop. So as Martí mentioned, I am going to be talking briefly about the radio-labeled approach to measure transformation rates of methylated sulfur compounds. So, okay. So you just saw a version of this graph that Martí showed. So I wanted to take a minute to acknowledge the contribution that the use of radio-labeled sulfur compounds has had for the past 25 years. So here you can see in light blue the publications using the radio-labeled approach. So about 25% of the total number of studies used radio tracers. And you can see this, that there were quite a few papers published in the early 2000s, another burst later on, but like Martí mentioned, a declining trend in the last years for both general publications of reduced sulfur cycling rates, but also for the radio tracer approach. But besides the importance of this approach to measure rates that can also be obtained by other approaches like we just saw, they have been very important to allow us to quantify processes that cannot be easily measured with other approaches, which we'll talk in a minute. And also the use of radioisotope has provided like key insights into the metabolic use of DMSP and DMS by microbes. Those are processes that now we take for granted, but at the time there were important discoveries. So I'm showing the same schematic that we know needs some updating, but it serves my purpose, which is to give you a quick picture of the measurements where radio-labeled tracers have been used to measure rates, focusing mostly on DMSP and DMS, which are the most commonly used. But keeping in mind that also labeled methionine and methanethiol have also been used, for example. So first we can measure the net loss of dissolved DMSP, which includes all the arrows that are coming out of this pool. We can also quantify the production of volatiles, but we can also specifically quantify the DMS produced from the consumed DMSP, which in the literature, that's normally referred as the DMS yield. We can also quantify the uptake and the assimilation into macromolecules by microbial cells, phytoplankton, and bacteria. And with radio-labeled DMS, we can measure, same as with DMSP, we can measure the net losses of DMS, which would be all the arrows that are coming out of DMS. And we can also quantify the fraction of that DMS that is either partially oxidized to DMSO, so we can obtain DMSO production rates from DMS, or that it is completely oxidized all the way to sulfate. And we can also quantify the fraction of that consumed DMS that is assimilated into macromolecules. So there are quite a few processes that we can measure with this approach. So almost all published studies have used sulfur-35 as the radioactive label, which it decays into chlorine and this beta particle. That beta particle can be easily measured through the interaction with what we call a scintillation cocktail. This cocktail is just a mixture of organics that when they react with this beta particle, it emits fluorescence, and that fluorescence is very easy to detect. And because the natural abundance of radioactive atoms in the environment is extremely low, even a small increase in their concentration becomes very easily detectable. And this makes this approach highly sensitive for measuring reaction rates. So this sulfur-35 has a half-life of 87 days. And this means that after that time, half of your initial radioactivity will have decayed. And it's not ideal, but it's workable. But what that means is that you can't have labeled DMSP sitting in your freezer for 2 years, because what will happen is that your specific activity will be very low after that time, and your stock is no longer useful. So the specific activity is telling you the amount of radioactivity that you have per mole or per millimole of your chemical. So the higher the specific activity, the less isotope you need to add to achieve a detectable signal. And then that means that you don't have to modify your in situ concentration much. And this is a very important aspect of this technique because you can truly make tracer additions. So for example, when we synthesize DMSP from methionine, we can obtain very high specific activity DMSP, and we can make amendments of less than 5 picomolar for months after we synthesize it. So, and we still have enough signal to detect changes during relatively short incubations. So let's assume we have this reactant A and a product B, and let's say we're measuring DMS consumption. So in this case, we basically inject our labeled DMS into the sample. And after we incubate the sample in our bottle, now we have both our labeled unreacted DMS and the labeled products from DMS consumption. So the activity now in that vial is the same as before. We don't know if the radioactivity is coming from A or from B. So one of the bases of this kind of radioactive work is that we always need a way to physically separate the labeled reactant from the labeled product. So that's when you have to be creative. If we assume that DMS was not transformed into any other gas, we can sparge out that unreacted DMS and quantify the amount of radioactivity that was left in the sample as non-volatile, which should be equal to the amount of labeled DMS that was consumed. So because both the DMS laws and dissolved DMSP laws, they follow first order kinetics, you can easily determine rate constants from the initial activity added and the activity of the product at the end of the incubation. So even though this equation here is showing you concentrations, we can replace those by activity of, in this case, of DMS at the beginning of the incubation and activity of the non-volatile products, just because of the proportionality between concentration and activity. So it's important to notice that what we are measuring are rate constants. So then, in order to obtain actual consumption rates in units of concentration per unit of time, we need to multiply that rate constant by the in situ concentration of the reactant. And we'll see now that that cannot be, sometimes it's not that simple. So this is just an example to show the potential for the technique. This is a depth profile of DMS consumption rates obtained in the allelotrophic Sargasso Sea, where you can see how we're able to determine very low rates of about 0.1 nanomolar per day of DMS consumption in surface water with high precision. And we also had enough signal to determine also the metabolic fate of that DMS. We were able to track that sulfur into the production of DMSO, of sulfate, and macromolecules. And here they are shown as percentage, but you can do some math and get production rates for those pools. But on the other hand, this is moving now into the MSPD turnover rates. This is a very interesting study by Lee et al., in which by doing side-by-side comparisons of the radiotracer versus the inhibitor approach that Rafel just explained, they found serious limitations in the use of radio-labeled DMSP to measure the dissolved DMSP turnover rates. So in this table, you can see the comparison of the rates obtained by the two approaches. And what they found was that the radio-labeled rates were up to 150 times higher than those obtained using the GBT inhibitor method. And what they concluded was that the problem was not in the determination of the rate constant, but the error was in what was considered bioavailable dissolved DMSP, which is the actual reactant pool. And the reason for that is that we normally define dissolved DMSP simply as the DMSP that goes through a GFF filter. And they found that in between 10 to 37% of that, of what we call dissolved DMSP, was actually associated to particles bigger than 0.2 microns, probably bacteria. So it was not truly bioavailable. And not only that, but also 40 to 99% of that dissolved DMSP was refractory to degradation. So again, not really bioavailable. And by comparing the two approaches, they came to the conclusion that only 1 to 60% of this operationally defined dissolved DMSP was bioavailable for transformation. This comparison was done in estuarine and coastal water, so maybe the overestimation of rates using the radio-labeled DMSP are not as bad in oceanic waters, but it's still an issue. And not only that, but if we are using this approach to quantify production rates of any of the products from dissolved DMSP transformation, for example, DMS production from DMSP, we are also carrying over that error. So we are also overestimating those other processes. So to sum up, this approach has the advantage of not altering significantly the in situ conditions, so being true tracer additions. They allow us to do small volume and relatively short incubations, which can be done under light or dark conditions. And we can also go beyond measuring net loss rates and quantify the metabolic fate of the sulfur. Or we could track the metabolic fate of the carbon, for example, if we were to use C14 labeled DMSP or DMS. And in terms of disadvantages, well, we do need to perform incubations, but that's true for all the approaches that we saw today. And we need to have accurate measurements of the reactant pool, which for DMSP can be tricky. And in terms of the logistics, the use of radioactive material, as you know, is subject to strict regulations, requires licenses, so that can complicate experiments, if they're even possible, because many research vessels do not even allow the use of radio-labeled chemicals. And also, and this is an important one, the labeled DMSP and DMS are not commercially available. so they need to be synthesized. And so far there have only been 2 labs that performed the synthesis. However, you might have seen this paper that was recently published in Marine Chemistry by Alexandra Smith et al. And where we explained the protocol for synthesizing labeled DMSP and DMS from methionine. And later I will paste a link in the comments for those who do not have access. So, and just to finish up, some open-ended questions, I guess. Do we need to further evaluate these approaches? How can we do that as a community? Do we think that these approaches are worth it? Do we want to facilitate, increase the use of radio tracers to measure rates? And again, how can we do that if that's the case? Are there other rates that are critical, and we can try to answer those with radio tracers that would increase our knowledge of the sulfur cycle and the carbon cycle. And that's all I have. Thank you for listening, and I'll stop sharing my screen now. I think I went a little bit over. I'm sorry.
Thanks, Daniela. This has been super interesting. Thanks a lot. And well, I guess we have time for a couple of very quick questions if someone wants to ask one. And so you can raise your hand or simply speak up. In the meantime, I will share my screen again. Yes, there's a question from Jacqueline.
Hi, Daniela. Nice overview. Did you ever try to compare the radio tracer with the stable isotope tracer method? I mean, of course there is a big difference in the concentration that you need, or for one and the other, but still, it would be interesting whether you get something like a similar specific rate or not.
Okay. I haven't done it, but I left Ron's lab a long time ago. But I am not aware of any side-by-side comparisons. But I guess it would be possible if the idea is to test the effect of having a higher concentration because of the nature of the stable isotope addition. I think that could be done relatively easily. You don't even need to actually measure that part. You just see the effect of having a higher concentration. So I guess indirectly we've done those kinds of tests, but not with that comparison in mind.
Well, it would probably only be interesting for the DMS consumption rate, because of course the benefit of the radio tracer is also that you can maybe trace it back in sulfate or other, I don't know. Do you actually do that, or do you only look at the DMS consumption rate, or also where it goes, right?
It depends on your question and how much work you want to put into it. But so you can do both, and you can do both from the same incubation that you are determining the rates. You can continue working with that same sample and trace it, follow it into other products.
Have you done that too?
Yeah.
Have you done that too?
Mm-hmm.
Or someone else, or?
No, both for DMSP and DMS I've done.
Yeah. I was just wondering whether you can, whether it's possible to measure sulfate, radio-labeled sulfate back or so, whether there's anybody, I don't know.
Well, yes. So sometimes, I mean, it depends. You can quantify it as barium precipitable. So you can assume that what you precipitate with barium is sulfate, but you can also check if that's actually the case later on. Yeah.
Thanks.
Great, thanks. So I think there was a question by John Todd. Do you still want to ask the question, John? Or the hand?
I don't know if I had a question. I think I was just fiddling.
Okay, sorry.
but actually, I can ask a question if you like. Did I miss questions because I changed rooms? Can I ask Rafel Simó a quick question if that's okay?
It's okay. Yeah.
Yeah. So he said Rafel, you said you did some incubations with glycine betaine. Do you monitor trimethylamine production as well, or can you see that when you do those incubations?
No, and that would have been fantastic. But at the time we did those incubations with GBT, that was meant to demonstrate, say, or to test whether it was inhibiting the MSP consumption somehow or uptake. And that was the only aim. So we did not do any effort to look at the products of any either GBT or DMS transformation into something else or whatever. No, so and we don't at that time. Well, we still don't have a method for trimethylamine or trimethylamine oxide analysis in our lab.
Okay. And I guess and swapping over to Danielle, that was a really nice talk as well. Sorry. So we have a lot of C14 labeled DMSP, but it's on the acrylic acid component, which we use routinely which we have used to trace the bugs that will eat it, and we have C13 labeled, which we'll use for stable isotope probing if we want to know what bugs are eating the DMSP, the acrylic acid component, as a carbon source. But we haven't really routinely gone for the sulfur component of it. I wondered if so you're focused on the DMS production, but what do you know about looking at the bugs that are eating the other component?
Is Dave Keever still here? I can pass that question to him.
So actually, hey everybody, this is Lei. I'm a postdoc with Dave. Actually, he just lost his electricity in his house, so he's kind of logged out. But he's trying to log in using his phone. I just answered his phone. So what was the question about acrylate? I just missed it. I was just answering his phone. So I heard there's a question about acrylate. I'm not sure if I can answer that or comment on that at all.
Well, John was asking whether acrylate, radiolabeled acrylate, is being used, and how is it being used to measure acrylate consumption. And you did that.
Yeah, we were trying to get that for the cruise to Maria back to 2018, but it was not allowed by the French Polynesia. And yeah, for my PhD, I basically study the phototechnology and consumption of acrylate and DMSP. We're really thinking about to get a C14 labeled acrylic acid to study the photolysis. But there were some reasons we just we couldn't get it, or the photolysis is really not happening, so we just didn't get it. The other way we can get acrylic acid with C14 labeling is just to get a C14 labeled DMSP, then just use base to generate it, the C14 labeled acrylic acid. But we haven't done that. I've graduated, I moved on to a new project, but I'm really interested in that aspect.